Welcome to Rad Onc Smart Review RadBio Edition. We're diving into the core molecular techniques, the ones that really underpin everything we understand about how radiation interacts with living cells.
Exactly. Think of this as, a focused tutoring session.
Right, designed to give you the fundamentals and hopefully some memorable tips as you gear up for your board exams.
Absolutely. We've talked about DNA damage and repair pathways before, haven't we?
We have.
So today, we're looking at the actual tools, the methods scientists use in the lab to see and measure and even manipulate the DNA, RNA, and proteins involved in all those critical processes.
So why are these so vital? I mean, for radiation biology and ultimately radiation oncology.
Well, because radiation's effects are, at their heart, molecular. We see the big picture effects in tissues and tumors, sure, but the initial events, they're things like DNA damage, kicking off repair pathways, hitting cell cycle checkpoints, changing gene expression. So to really get a handle on why some tumors are resistant or why normal tissues react the way they do, you absolutely have to look at this molecular level.
So these techniques are kind of like our molecular toolkit. They let us actually count the damage, see the repair spots, measure how genes switch on or off.
Or even directly change genes to see what happens.
Yeah, test their role in radiation response.
Precisely. They give us the hard data, the evidence that informs our models, our understanding, and down the line, our clinical strategies.
Okay, let's jump in. Starting with the workhorses for nucleic acids, DNA, and RNA. First up, polymerase chain reaction, PCR. Everyone knows it, right?
Oh, yeah. PCR is absolutely fundamental. Its real power is taking just a tiny, tiny bit of DNA, sometimes literally a single molecule, and making millions, even billions of copies of a specific bit you're interested in.
The molecular photocopier analogy works well here, doesn't it? Pick a page, hit copy, repeat exponentially.
Exactly. And like any copier, you need the right stuff. You need your DNA template that's the starting material with the sequence you want to copy.
Your original document.
Then primers. These are short, single-stranded bits of DNA. They're designed to stick, or anneal, just before and just after the region you want to copy. They basically tell the polymerase where to start and stop.
Like highlighting the bit you want copied. Got it.
Yep. You also need a special enzyme, Taq polymerase. It's named after Thermus aquaticus, a bacterium found in hot springs.
Ah, so it's heat stable, thermostable.
Right. It can handle the high temperatures needed for PCR without breaking down.
Yeah.
It's the enzyme that actually builds the new DNA strands.
A heat-proof builder enzyme. What else?
The building blocks themselves.
Oh.
The deoxynucleotide triphosphates, or DNTPs, DTP, DGTP, DCTP, and DTTP.
AGCT bases.
And a buffer solution, just to keep the chemical environment right for Taq to do its best work.
Okay, so the process involves temperature cycling. What are those key steps?
3 main steps, repeated maybe 25 to 35 times. First, denaturation. Heat it up, usually 94 to 98 degrees Celsius. This high heat breaks the hydrogen bonds holding the DNA double helix together.
Separates the strands, unzips it.
Exactly. Then annealing. Cool it down, typically 50 to 65 degrees Celsius. This lower temperature allows those primers you added to find and bind to their specific complementary sequences on the now single-stranded templates.
Primers stick on.
Finally, extension or elongation. You raise the temperature again, usually to about 72 degrees Celsius. This is the sweet spot for Taq polymerase. It latches onto the primer and starts adding DNPPs, synthesizing a new DNA strand complementary to the template.
And the crucial part is that the DNA made in one cycle becomes a template for the next.
Precisely. So you get this exponential amplification. Theoretically, the amount of your target DNA doubles with every cycle. That's why it's so powerful you can detect tiny amounts of DNA or make enough for other analyses, like sequencing.
That exponential growth is the magic.
Okay, let's shift gears to another set of foundational techniques, the blotting methods. Used for detecting specific molecules, DNA, RNA, or protein. Right, blotting techniques are classic. The basic idea is you separate molecules by size using gel LEK trophoresis, then you transfer them, that's the blotting part, onto a solid membrane, and then you use a specific probe or antibody to find the one you're looking for in that complex mixture.
And there's that super helpful mnemonic everyone needs to know, snowdropy.
Oh, absolutely essential. You got to know this for boards. Snowdropy. Southern detects DNA.
Snow D.
Northern detects RNA.
Drop R. Wait, no. Snow D. Northern and RNA. Okay.
Southern DNA, Northern RNA, and Western.
W protein. Snow D drop P. Got it. Okay, let's break down Southern first. Detects DNA, named after Edwin Southern. How's it work?
Okay, you start by isolating DNA. Often you'll cut it into smaller pieces using restriction enzymes. These enzymes cut DNA only at specific sequences.
Right.
Then you separate these fragments by size using agarose gel electrophoresis. Bigger pieces move slower, smaller pieces move faster.
Okay.
Before you transfer, you usually treat the gel with an alkaline solution to denature the DNA, making it single-stranded.
Why single-stranded?
So your probe can bind later. You need complementarity. Then you transfer the single-stranded DNA pattern from the gel onto a membrane nitrocellulose or nylon usually. This transfer, the blotting, is often done by capillary action.
Like making a print of the gel pattern onto the membrane?
Kinda, yeah. The DNA sticks to the membrane in the same pattern it had in the gel. Then comes high-H-brid IZAAY shun. You incubate the membrane with a labeled probe. This probe is a piece of DNA or RNA that's complementary to the specific DNA sequence you're hunting for.
So it only sticks to your target DNA band.
Exactly. It hybridizes only where its complementary sequence is located on the membrane. Finally, detection. You visualize where the probe bound. If it's radioactive, you use AW to ATD-OG RUFI. More commonly now, probes have tags that allow detection via chemiluminescence or fluorescence. The band tells you the size of the DNA fragment containing your target.
And historically used for things like finding specific genes, checking for rearrangements, or RFLP analysis, which we'll touch on.
Exactly. Gene detection, structure analysis.
Okay, moving to N in Snowdrop, Northern blot. For RNA, similar process.
Very similar principle, but your target is RNA, typically messenger RNA, mRNA. You extract RNA, separate it by size on a gel, often under denaturing conditions to keep the RNA from folding up, weirdly transfer to a membrane, hybridize with a labeled probe, can be DNA or RNA, and detect.
What does Northern tell you that Southern doesn't?
So Southern looks at the DNA blueprint itself as the gene there. Is it rearranged? Northern looks at gene expression. By detecting mRNA, you see if a gene is actually being transcribed into RNA. How much mRNA is there? What size is it? Are there different versions from alternative splicing? It's a direct measure of gene activity at the RNA level.
Makes sense. And finally, W for Western blot. Detecting protein. What's different here?
Okay, target is protein. First, extract proteins from your cells or tissue. A key step is you usually denature the proteins, often by boiling them with SDS, that's sodium dodecyl sulfate.
Right. SDS does two things, doesn't it? Unfolds proteins and gives them a negative charge.
Exactly. It coats them with a uniform negative charge, so when you run them on a gel, they separate primarily based on their size, not their native charge or shape.
Okay, so you run them on SDS page polyacrylamide gel electrophoresis.
Correct. Proteins separate by size. Then, just like the others, you transfer the proteins from the gel onto a membrane, typically nitrocellulose or PVDF. Now, a crucial step unique to Westerns is blocking. You incubate the membrane with a generic protein solution, like milk protein or BSA, bovine serum albumin.
Why block?
To prevent your antibodies from just sticking randomly all over the membrane surface. You want them to bind only to your protein of interest. Blocking covers up the non-specific binding sites.
Got it. So antibodies only find their specific target protein band.
Precisely. Then you add your primary antibody. This one is specifically designed to recognize and bind only to your target protein. You wash away any unbound primary antibody, then you add a secondary antibody.
And the secondary binds to the primary.
Yes, and the secondary antibody has a tag attached to it, often an enzyme like HRP, horseradish peroxidase, or a fluorescent dye. If it's an enzyme like HRP, you then add a chemical substrate that the enzyme reacts with to produce a detectable signal, like light, chemiluminescence, or a colored spot.
And you detect that signal maybe on film or with a digital imager, the stronger the signal.
The more protein you likely have. Westerns are workhorses. You use them to confirm if a protein is present, see if its levels go up or down after, say, radiation, or even detect specific modifications like phosphorylation using special antibodies.
Okay, super important. Remember, snow dewdroppy, southern DNA, northern RNA, western protein. Nail that down.
Definitely board favorite.
All right, moving beyond just detecting molecules, sometimes you need the exact sequence, the ATCG order, DNA sequencing. We started with Sanger.
The classic method.
And now we have the massive power of next-generation sequencing, NGS. Tell us about Sanger first, the chain termination method.
Right, developed by Fred Sanger, a huge breakthrough. You can think of it like figuring out a sentence one letter at a time by creating fragments that stop at every instance of a specific letter.
Building the sequence piece by piece.
The key is using DNA polymerase to copy your DNA template, but you include a small amount of special nucleotides called dideoxy nucleotides, or DDNTPs.
The terminators.
Exactly. They lack the chemical group needed to add the next base. So when a DDNTP gets incorporated by the polymerase, the chain stops growing right there.
Okay.
In the original method, you'd run 4 separate reactions, each with all the regular DNTPs, plus a small amount of one specific DDNTP. DDATP, DDTTP, DDCTP, or ADDTP. This generates a collection of DNA fragments of different lengths, each ending with a specific known base. You'd separate these fragments by size on a high-resolution gel.
And read the sequence from the ladder of bands.
Right. Modern Sanger uses fluorescent dyes, a different color for each DDNTP, all in one reaction tube. Then you separate the fragments by size using capillary electrophoresis. A laser detects the color of the fluorescent tag on the last base of each fragment as it passes by, and a computer reads the sequence.
And Sanger gives you pretty long, accurate reads, maybe hundreds to 1,000 base pairs. Great for specific things like PCR products or checking mutations.
Exactly. It's still the gold standard for accuracy on a smaller scale, but it's lower throughput and more expensive per base compared to
Next generation sequencing, NGS. Massively parallel sequencing. This really changed the game.
absolutely revolutionized genomics. Instead of sequencing one DNA molecule at a time, NGS platforms sequence millions or even billions of DNA fragments simultaneously. Illumina is a very common platform, but there are others like Ion Torrent, PacBio, Oxford Nanopore. The core idea is this massive parallelization.
How does that actually work? Take the Illumina approach, for example.
Okay, Illumina uses sequencing by synthesis. First, you prepare a library. You fragment your DNA sample into smaller pieces and ligate, or attach, short-known DNA sequences called adapters to the ends of each fragment.
Okay, adapters on the ends.
These adapter-ligated fragments are then flowed onto a special surface called a flow cell, which has complementary adapters attached. The fragments bind. Then, each bound fragment is amplified in place through a process called bridge amplification, creating a dense cluster of identical DNA molecules. You end up with millions of these clusters scattered across the flow cell.
Millions of little identical DNA colonies.
Exactly. Then the sequencing itself begins. The flow cell is flooded with DNA polymerase and special fluorescently labeled nucleotides. These nucleotides have a reversible terminator group, meaning only one base can be added at a time.
Ah, like Sanger's DDNTPs, but reversible.
All right, so in each cycle, one fluorescently labeled nucleotide is added to the growing strand in every cluster. A laser excites the dye, and a camera captures the color emitted from each cluster. The color tells you which base, A, T, C, or G, was added. Then, the fluorescent dye and the terminator group are chemically cleaved off.
Allowing the next cycle to start.
Yes, this cycle repeats at a base, image the color, cleave the dye terminator over and over, maybe hundreds of times, simultaneously for all million clusters.
Incredible. So you generate millions of short sequence reads.
Yeah, typically short reads, maybe 50 to 300 base pairs long. The computer collects all this image data and translates it into sequences. Then comes the heavy bioinformatics part, aligning these millions of short reads to a reference genome, like putting together a giant jigsaw puzzle or assembling them from scratch if you don't have a reference.
The sheer amount of data must be immense.
It is. But this high throughput dramatically lowered the cost and time for sequencing. It's enabled huge projects: whole genome sequencing, WGS, exome sequencing, just the protein coding bits, RNA-seq to look at all the RNA transcripts.
Transcriptomes.
Right. Targeted panels focusing on specific cancer genes, studying microbial communities, metagenomics, how DNA is packaged, epigenomics. It's just opened up so many doors.
Before NGS really took off, another way to look at DNA variation was RFLP analysis. Restriction fragment length, PAH-Lee MORFism.
RFLP, yeah. An older technique, historically important for genetic mapping, finding disease genes, early DNA fingerprinting. The concept is still relevant, though. It hinges on natural variations in DNA sequences between individuals.
Variations that might create or destroy a cutting site for a restriction enzyme.
Exactly. A single base change can sometimes abolish a site where a specific restriction enzyme normally cuts, or create a new one. So if you take DNA from different individuals, digest it with that enzyme, the resulting DNA fragments will be different lengths if they have that sequence variation, that polymorphism.
Okay.
You'd isolate DNA, cut it with the enzyme, separate the fragments by size on an agarose gel, and then do a southern blot using a probe for the region you're interested in.
And you'd see different patterns of bands on the southern blot depending on the person's RFLP genotype.
Yes, different fragment lengths mean different banding patterns. It was a key way to track genetic markers linked to diseases before we could easily sequence things. While sequencing is much more powerful now, understanding how sequence changes affect restriction sites is still a useful concept.
Definitely. Okay, let's pivot now to techniques specifically focused on detecting DNA damage, particularly the kind caused by radiation. The Comet assay and gamma H2AX foci staining are really central here.
Absolutely critical for radiobiology. The Comet assay, or single-cell gel electrophoresis, is super sensitive for detecting DNA strand breaks within individual cells.
And it gives that really cool visual, the comet shape under the microscope.
It does. The principle is pretty neat. Broken DNA is less structurally constrained than intact supercoiled DNA within the nucleus.
Oh.
So if you embed cells in a gel and apply an electric field, the broken bits can migrate out of the main nuclear mass.
Okay, how do you do it?
You embed single cells in a thin layer of agarose gel on a slide. Then you lyse the cells using detergents and high salt to remove membranes and most proteins, leaving behind the DNA packed in its basic structure, the nucleoid. Then you apply an electric current, electrophoresis. The intact DNA mostly stays put in the head of the comet.
While the damaged DNA streams out.
Exactly. The broken fragments and relaxed loops get pulled towards the positive electrode, forming the tail. After electrophoresis, you stain the DNA with fluorescent dye, like ethidium bromide or SYBR green, and look at it under a fluorescence microscope.
And the comet's tail tells you about the damage level.
Yes, more damage means more DNA migrates into the tail, making the tail longer and brighter. You can quantify this with image analysis measure tail length, the percentage of total DNA that's in the tail, or something called the tail moment, which combines length and intensity. It gives you a quantitative measure of DNA breaks per cell.
Now, I know there are different pH versions, neutral and alkaline.
Very important distinction. The neutral Comet assay, run at neutral pH, primarily detects double-strand breaks, DSBs. It's thought that only DSBs cause enough relaxation for significant migration under neutral conditions.
DSBs being the really critical lesion from radiation generally.
Often considered the most lethal, yes. The alkaline Comet assay, however, is run at very high pH, usually above 13. This denatures the DNA, separating the strands. Now, single-strand breaks, SSBs, can also migrate out. DSBs will show up too because they effectively become two SSBs when denatured. Plus, the high pH can reveal alkali labile sites, damaged bases that break down into strand breaks under alkaline conditions.
So the alkaline version is more sensitive overall, detects more types of damage?
Yes, it detects SSBs, DSBs, and those alkali labile sites. It's generally more sensitive and can detect damage from very low radiation doses, even down around 1 centigrade.
Wow. So it's mainly a research tool for measuring damage induction and repair kinetics.
Exactly. You can measure how much damage appears right after exposure and then track how the comet tails shrink over time as the cells repair the breaks. Invaluable in radiobiology and toxicology.
Okay, another key technique, especially for specifically visualizing DSBs, is gamma H2AX foci staining. Gamma H2AX.
Yes, this is a standard and extremely useful technique. It lets you see the actual sites of DSBs within the cell nucleus. It relies on a rapid cellular response to the break itself.
What is that response?
When a DSB occurs, specific protein kinases enzymes like ATM, ATR, and DNA-PK quickly add a phosphate group to a particular histone protein called H2AX, specifically at serine residue 139.
Histones are the proteins DNA wraps around, right? Part of chromatin structure.
Correct. This phosphorylated form of H2AX is called gamma H2AX, and it doesn't just happen right at the break. It spreads along the chromatin for quite a distance, potentially megabases, flanking the break site. It acts like a biochemical flag, signaling damage here.
And you use antibodies to see these flags.
Precisely. It's an immunofluorescence method. You treat your cells, maybe radiate them, then fix them to preserve their structure, and permeabilize the cell membrane so antibodies can get in. Then you incubate with a primary antibody that specifically recognizes the gamma H2AX modification.
Okay.
Wash away unbound antibody, then add a secondary antibody. The secondary antibody binds to the primary one and has a fluorescent dye attached. Common ones are FITC for green fluorescence or Alexafluor 594 for red.
Then you look under a fluorescence microscope.
And you see these distinct bright spots, or foci, within the nucleus. Each focus generally corresponds to the location of one DSB that has recruited gamma H2AX.
So you can actually count the dots per cell to quantify the number of DSBs.
Yes, it's a direct quantitative measure. You can count the average number of foci per cell right after radiation to measure the initial damage induction, and then you can look at later time points and see the number of foci decrease as the DSBs get repaired.
Right.
This allows you to study DSB repair kinetics.
Very powerful. Used a lot in research.
Hugely. Quantifying DSB induction and repair, comparing radiosensitivity between cell lines, testing radiosensitizers, It's also being investigated as a potential clinical biodosimeter, maybe to estimate radiation dose received in an accident, or even predict how sensitive a patient's normal tissues might be to radiotherapy.
So, quick recap. Comet assay gives a broader measure of strand breaks, especially the alkaline version, while gamma H2AX is highly specific for visualizing and counting DSBs.
That's the key takeaway. Both are incredibly valuable.
Okay, let's shift to techniques that look at how proteins interact with DNA to control genes. Chromatin immunoprecipitation, or CHIP, is a major one here.
CHIP is a technique you use when you want to know where a specific protein is physically bound to the DNA inside the cell in its natural chromatin environment. Think transcription factors binding to promoters or repair proteins localizing to damaged DNA. It maps the protein's address on the genome.
How do you actually isolate the DNA bits that a specific protein is sitting on?
It's clever. You use an antibody against your protein of interest to essentially pull down that protein. And any DNA it's currently attached to comes along for the ride. The first step is usually cross-linking. You treat the cells with formaldehyde.
Formaldehyde?
Yeah, it creates chemical links, covalent bonds between proteins and nearby DNA and other proteins. This effectively freezes the protein-DNA interactions as they were happening in the cell.
Locks them together.
Exactly. Then you need to break the chromatin into smaller, manageable fragments. This is usually done by sonication using sound waves or enzymatic digestion. You want fragments of a few hundred base pairs, ideally. Some of these fragments will have your cross-linked protein of interest attached.
Okay, fragmented chromatin, protein still attached.
Next comes the core step, immunoprecipitation, the IP in CHIP. You add an antibody that specifically binds to your protein target, say, an antibody against HIF-1 alpha if you're studying hypoxia. Then you use special beads, often magnetic or agarose beads, coated with something that binds the antibody, like protein A or G, to capture the antibody-protein DNA complexes.
So the beads grab the antibody, which is grab the protein, which is stuck to the DNA.
Right. Then you wash away everything that didn't bind all the chromatin fragments that didn't have your specific protein attached.
Purifying the complexes.
Yes. Now you have isolated the DNA fragments that were associated with your protein. You reverse the cross-links, usually by heating, and digest away the proteins using proteinase K. What you're left with is purified DNA, the specific sequences your protein was bound to.
And then you need to figure out what those DNA sequences are. How do you do that?
Several ways. If you have a specific hypothesis, like, does my protein bind to the promoter of gene X? You can use PCR with primers designed for that promoter region. This is CHIP PCR. If the PCR works on your CHIP DNA, it means the protein was likely bound there.
What if you want a broader view?
You could use ChIP-ChIP, which involves hybridizing the CHIP DNA to a microarray containing probes for many genomic regions. But the most common and powerful method now is ChIP-seq. You take all the DNA fragments you purified from the CHIP experiment and sequence them using next-generation sequencing.
Ah, using NGS again.
Yes. Then you map all those sequence reads back to the genome. The regions where lots of reads pile up indicate the sites where your protein was bound. This gives you a genome-wide map of binding sites.
So you could map exactly where a repair protein goes after radiation, or identify all the genes directly regulated by a specific transcription factor under certain conditions, like hypoxia.
Exactly. It's incredibly powerful for understanding gene regulation and protein localization. Another key technique for studying gene regulation, specifically the activity of promoters or enhancers, is the reporter gene assay.
Right. You used the dimmer switch analogy earlier. The promoter is the switch. The reporter gene is the light bulb.
That's a great way to put it. The idea is you link a piece of DNA that you think regulates gene expression like a promoter to a gene whose product is really easy to detect and measure. This is the reporter gene. The amount or activity of the reporter protein produced then serves as a readout or a report of how active your regulatory DNA sequence is.
So you build a special piece of DNA, a construct.
Yes, using molecular cloning techniques. You basically cut and paste DNA pieces together. You create a plasmid vector where your regulatory DNA sequence, say the promoter of the VEGF gene, is placed just upstream of the coding sequence for a reporter gene.
And what makes a good reporter gene?
It needs to be easily measurable and preferably not naturally present in the cells you're using. Luciferase, often from fireflies or sea pansies, is very popular. It catalyzes a reaction that produces light when you add its substrate, luciferin. You measure the light with a luminometer, it's extremely sensitive.
Okay.
Green fluorescent protein, GFP, from jellyfish is another big one, along with its colorful cousins, YFP, yellow, RFP, red, etc. GFP naturally glows green when you shine blue or UV light on it. You can see it in living cells with a microscope or quantify the fluorescence. Beta-galactosidase, last Z, can turn a colorless substrate blue. C-galenicol acetyltransferase is an older one.
So you make this plasmid construct, then what?
You introduce it into the cells you want to study. This is called transfection. The cell's machinery will then read the construct. If your promoter sequence is active under the cell's current conditions, it will drive transcription of the reporter gene, leading to reporter protein being made.
Then you apply your experimental treatment.
Exactly. You treat the transfected cells, maybe irradiate them, put them in low oxygen, add a drug, overexpress another gene, whatever you're investigating. Then you measure the reporter signal.
How?
Depends on the reporter. For luciferase, you release the cells, add luciferin, and measure light output. For GFP, you might measure fluorescence intensity with a plate reader or flow cytometer.
And if the signal goes up after treatment, it means your regulatory sequence was activated.
Precisely. More light, more fluorescence means the promoter or enhancer became more active, driving more reporter expression. If the signal goes down, it was repressed. It's a functional readout of regulatory element activity.
Very useful for testing hypotheses about gene control now. Let's talk about CRISPR. CRISPR-Cas9 gene editing, this has been absolutely transformative.
Oh, completely. CRISPR-Cas9 is like having programmable molecular scissors for the genome. It allows for incredibly precise, targeted modifications to DNA sequences within living cells.
What are the key parts of the system?
Two core components you need to get into the cell. First, the Cas9 protein. This is an enzyme, specifically a nuclease, that cuts double-stranded DNA.
The scissors part.
Exactly. Second, you need a guide RNA, or gRNA. This is a synthetic RNA molecule that you design. Part of it binds to the Cas9 protein, and another part, typically about 20 nucleotides long, is designed to be perfectly complementary to the specific DNA sequence in the genome where you want Cas9 to cut.
So the gRNA is the targeting system, guiding the Cas9 scissors to the right address.
Precisely. There's one more small requirement: the Cas9 enzyme won't cut just anywhere. It needs to recognize a short, specific DNA sequence called the PAM sequence, protospacer adjacent motif, located immediately next to the target site where the gRNA binds. Different Cas enzymes have different PAM requirements, but the common Streptococcus pyogenes at the Cas9 needs an NG sequence.
Okay, so you deliver Cas9 protein, or the gene encoding it, and the custom-designed gRNA into your cells. What happens?
The gRNA forms a complex with the Cas9 protein. This complex then scans along the DNA looking for a match. When the gRNA finds its complementary target sequence on the DNA and the correct PAM sequence is right next door, the Cas9 enzyme locks on and cuts both strands of the DNA, creating a double-strand break, DSB, at that specific location.
A precisely targeted DSB, and then the cell tries to fix it.
Exactly. The cell detects the DSB and activates its DNA repair machinery. And how the cell repairs that break is what leads to the gene editing outcome. There are two main pathways.
Okay, what's the first one?
The most common pathway, kind of the cell's default quick fix, is non-homologous end joining, or NHEJ. It basically just tries to stick the two broken ends back together as quickly as possible. But NHEJ is often sloppy. It's error-prone.
Meaning?
Meaning it frequently introduces small errors right at the break site, typically small insertions or deletions of a few base pairs. These are called indels.
And if these indels happen within the protein coding part of a gene?
They can shift the reading frame, messing up the entire downstream amino acid sequence, or introduce a premature stop signal. This usually results in a non-functional protein. So if your goal is to knock out or disable a gene, you rely on CRISPR to make the cut and NHEJ to make the inactivating mistake during repair.
That's the most common outcome for gene disruption. What's the other repair pathway?
Homology-directed repair, or HDR. This pathway is much more precise. It uses an undamaged DNA sequence as a template to accurately repair the break. Normally, it uses the sister chromatid if the cell is dividing.
But you can trick it.
Yes. If you provide an artificial DNA repair template, along with the Cas9 and gRNA, the cell can use that template during HDR. This template is designed to have sequences that match the DNA flanking the break site, the homology arms, but in between, you can include whatever change you want, maybe correcting a disease-causing mutation, inserting a specific sequence like a tag, or making a precise single base change.
So HDR allows for precise gene correction or insertion, rather than just knockout.
Correct. It's the pathway for making specific intended edits. However, HDR is generally much less efficient than NHEJ, and it mainly works in cells that are actively dividing during X and G2 phases of the cell cycle.
Still, the potential applications are huge.
Yeah.
Gene knockout, gene correction, inserting tags, even using modified dead Cas9 to turn genes on or off without cutting.
Absolutely.
Wow.
It's revolutionized how we can create cell or animal models of disease, test gene function, screen for drug targets. A really powerful tool in the radiobiology toolkit as well for studying genes involved in radiation response.
So bringing it all together, how do these diverse molecular techniques actually impact clinical decision-making in radiation oncology or guide research that leads there?
They're really the foundation for progress. While many are primarily research tools right now, the knowledge they generate directly fuels clinical advances. Take PCR variations like RT-qPCR are used constantly in labs to see how gene expression changes after radiation, identifying response pathways. Clinically, PCR detects viral DNA or specific cancer mutations that might predict response to targeted drugs, which increasingly intersects with radiotherapy.
Right, and blotting, especially Westerns.
Westerns are workhorses in the lab. You might see a study showing a key DNA repair protein level goes up in resistant cells after radiation, suggesting it's a potential target to overcome resistance. That research finding, based on a Western blot, could spark development of a drug targeting that protein to use alongside radiation.
And NGS is clearly making inroads into the clinic.
Definitely. Tumor sequencing, whether it's large panels, exomes, or even whole genomes, is becoming more common. Finding specific mutations like an EGFR or KRAS, or amplifications or fusions, can guide systemic therapy choices, which definitely influences radiation planning and goals. RNA-seq gives a snapshot of the tumor's active biology, potentially revealing vulnerabilities. It's all moving towards more personalized radiotherapy based on the specific molecular features of that patient's tumor.
Even older techniques like RFLP had a clinical impact eventually.
For sure. RFLP was crucial in the early days of mapping genes like BRCA1 and BRCA2 associated with hereditary cancer syndromes. Identifying those genes has had massive clinical implications for screening, prevention, and treatment choices, including radiation decisions.
What about the damage detection assays, Comet and gamma H2AX? Are they purely research?
Mostly research tools now, but incredibly important research. They let us quantify damage and repair, evaluate potential radiosensitizers or protectors before they get near patients, compare different radiation types or fractionation schemes. But there's definite interest in clinical applications. Gamma H2AX is being actively explored as a potential clinical biodosimeter, maybe measure foci in lymphocytes after treatment to gauge normal tissue dose or predict toxicity. It's not standard practice yet, but the potential is there.
And chip and reporter assays, still mainly lab-based.
Yes, lab workhorses. CHIP helps identify the proteins and pathways controlling genes involved in radioresistance or tumor progression. Reporter assays let researchers quickly test if a specific treatment turns critical genes, like apoptosis genes, on or off. This fundamental research points towards new therapeutic strategies and targets.
And CRISPR, still mostly research, but with huge future potential.
Exactly. Right now, CRISPR is revolutionizing research by allowing precise testing of gene function. Does knocking out gene Y make cells more sensitive to radiation? CRISPR lets you answer that directly. This rapidly accelerates the identification and validation of new targets that could eventually lead to new therapies combined with radiation.
Okay, great overview. Let's try to boil some of this down into essential clinical pearls, key takeaways for boards and practice.
That's good. Pearl number 1, you absolutely must know the snowdrop mnemonic, southernity, northern RNA, western ET protein. Remember the core idea: separation by size on a gel, transfer to a membrane, detection with a specific probe or antibody.
Got it. Pearl 2.
Pearl number 2, the Comet assay. It detects DNA strand breaks in individual cells. Remember the key difference: alkaline conditions detect SSBs, DSBs, and alkali labile sites, more sensitive, broader range, while neutral conditions primarily detect DSBs. The Comet tail gives you a quantitative measure of damage.
Okay, Pearl 3.
Pearl number 3, gamma H2AX foci. These specifically mark the sites of DNA double-strand breaks. It works by detecting the phosphorylated form of histone H2AX at serine 139 via immunofluorescence. Counting the foci in the nucleus gives you a direct quantification of DSBs and lets you track repair over time. A cornerstone assay in radiobiology.
Excellent. And Pearl 4.
Pearl number 4, CRISPR-Cas9 gene editing. It's the go-to for precise genome modification. The guide RNA, gRNA, directs the Cas9 nucleus to make a DSB at a specific target site. Remember the two main repair outcomes. NHEJ, non-homologous end joining, is error-prone and often leads to gene knockout via indels. HDR, homology-directed repair, if you provide a template, allows for precise edits or insertions.
Perfect. Those 4 pearls cover some really high-yield concepts.
Absolutely. Keep those straight in your head.
Ready for a quick board blitz? Test that recall.
Let's do it. Hit me.
Question 1. A researcher wants to determine if a specific transcription factor binds to the promoter of a target gene in irradiated cells. Which molecular technique would be most appropriate? A. Southern blot B, reporter gene assay C, CRISPR-Cas9D, chromatin immunoprecipitation. CHIP.
Okay. Transcription factor binding to DNA. That screams CHIP. D. Chromatin immunoprecipitation. CHIP is designed specifically to find protein-DNA interactions in the cell.
Correct. Question 2. A scientist observes that cells exposed to radiation show increased expression of a specific mRNA. Which blotting technique would confirm this finding by directly detecting and quantifying that mRNA? A. Southern blot B, Western blot C, Northern blot D, Eastern blot.
Increase mRNA. mRNA is RNA. It's noted to drop. N is for northern RNA, so C, northern blot.
Correct again. Northern detects RNA. Question 3. A highly sensitive method for detecting DNA strand breaks in individual cells, where damaged DNA migrates out of the nucleus forming a tail during electrophoresis, is known as the A. Gamma H2AX foci assay B, filter elution assay C, Comet assay D, RFLP analysis.
The tail is the giveaway. That's the Comet assay, C.
Perfect. Question 4. To specifically knock out the function of gene X in a cancer cell line to study its role in radioresistance, the most precise and currently favored gene editing tool would be A. Site-directed mutagenesis using PCRB, RNA interference, RNA, C. CRISPR-Cas9D, Crelox recombination.
Precise gene editing, specifically knockout. That's CRISPR-Cas9, C. RNAi knocks down expression temporarily, doesn't edit the gene itself. Crelox is powerful, but typically for larger-scale deletions or conditional knockouts. CRISPR is the tool for targeted knockout via DSB and NHEJ.
Excellent. You aced the blitz. Now, just a quick thought on how things evolve. We mentioned how NGS has really taken over from older methods like RFLP for large-scale stuff.
Absolutely. Technology doesn't stand still. High-throughput sequencing changed everything for genomics. And even blotting, while still used, is less common for initial discovery compared to RNA-seq for gene expression, for example.
And CRISPR is still rapidly advancing, right? New versions, better delivery.
Oh yeah, the field is moving incredibly fast, improving efficiency, specificity, expanding the types of edits possible. The potential for research is immense, and people are definitely working hard on translating it safely and effectively to the clinic, although that's still mostly in the future for cancer therapy applications combined with radiation.
So wrapping up, we've covered a lot of ground today. These essential molecular techniques really form the bedrock of modern radiobiology.
We hit PCR for amplification, the snowdrop blots, southern for DNA, northern for RNA, western for protein, the sequencing evolution from Sanger to NGS, and the historical context of RFLP.
Then we looked at DNA damage detection with the Comet assay and the specific DSB marker, gamma H2AX foci. Plus tools for gene regulation like SHIP for protein binding and reporter assays for promoter activity.
And capped it off with the revolutionary gene editing technology, CRISPR-Cas9. Understanding these tools, what they do, how they work, what they tell us is truly fundamental for anyone studying radiation biology or preparing for board exams. Keep putting in the effort, reviewing these concepts. You really can master this material.
Great advice. Next time, in episode Radiation Biology 32, we'll explore molecular imaging, CT, and PET in radiation oncology, really bridging that gap from the lab bench to clinical imaging.
Looking forward to it.
Remember, you can find complete practice, oral boards, and other resources over at radoncsmartlearn.com. And please subscribe to Rad Onc Smart Review so you don't miss our next discussion. Thanks so much for tuning in.